Hey everybody, my name is Jason Coleman. I'm a clinical application scientist here at Precision NanoSystems. Like Adam had mentioned, I'm going to be talking to you today about scale up and GMP vaccine production. I guess kind of the first question to look at and ask here is when we're developing a GMP vaccine, who is our ultimate patient population going to be? And as we live now in the world of COVID, we obviously know that it's a population of the world. And then how do you then develop a vaccine for the world from a manufacturing and production standpoint? So, both Jagbir and Adam have talked about self-amplifying RNA and the benefits of that. And in terms of producing a worldwide vaccine, the biggest benefit of that is you can produce a highly potent dose. And from a manufacturing standpoint, the benefits of that become you need a smaller manufacturing footprint, which makes production on a relatively smaller scale easier also. And also here's as a consider alternative approaches to multi-dose. Basically, when you're producing your vaccine and filling your vials and bags, you could have the potential to put more than one dose in one bag that can be redistributed at the injection site. And then, another goal here, which we've seen, a lot of the cold chain storage requirements with LNP vaccines, requirements of -8 or -20 can be difficult when distributing to globally. So the ultimate goal is to develop something that doesn't need cold chain storage, produce a lyophilized RNA vaccine. But obviously, the challenge then adds to your development time. So kind of to potentially compensate for that. Another benefit, something you want to do is have a manufacturing platform which can enable decentralized vaccines, so that basically the manufacturing can be done more regionally to where it's going to be administered. And this is where a big advantage of our GMP nanoassembler technology comes into play. So if we take a look at what is the ultimate production process for an RNA-LNP, this is basically the overall workflow here. So we have the microfluidic technology. Here is what we call our next-gen technology. So this architecture here for the production of making the particles, this architecture gives you the advantage in that you can make particles at a variety of scale, from very small scales in the lab all the way up to GMP production. And then also on the equipment you can do inline dilution, because as we all know, when you're making an LNP, there's ethanol as part of your product, so you need to reduce the ethanol to help stabilize your LNPs. And then ultimately, for downstream processing, typically tangential fluid filtration is done for purposes of then removing the ethanol and also putting your LNPs whenever your final storage or cryo buffer is going to be. Finally, followed by sterile filtration and your fill and finish process to where you have your final drug product. So what we have at Precision NanoSystems is the ultimate, the full range of equipment platform. So you can do all of this development work ultimately yourselves. And then, we can obviously help out as needed with that. So, on the Ignite, or Jagbir talked about, you can do your initial kind of formulation, preclinical development, where you identified different quality attributes of your formulation. And then with the Blaze, you can start to do your actual process development work. So you can see you can make up to 10-liter formulation size on there, and you can do this in R&D settings. So you have minimal waste. You can start to develop what that downstream, what that TFF process is going to be, and then finally you can transfer your process for GMP manufacturing on our GMP system with flow rates up to 200 mils per minute, which in a 4-hour time frame will give you the ability to make about a 50-liter formulation. So what we've talked about today, we heard some talks about going from development to the clinical. So I'm going to focus more on the clinical here. So in the development stage of the process with Jagbir's talk, that's the stage where you want to start to identify what are your critical quality attributes, and then also what are your critical process parameters, because that's what you're going to want to look at as you make any changes to your process. How are those effective? And then, as with any development program, what's also extremely critical is the analytical piece of it, which gets back to what Adam's talk was, where really anytime you need to have good analytical methods so that so you want to know how any changes that you make through formulation or through your process optimization, how those are going to affect your product, and you need assays in order to do that. And then also, I can't kind of emphasize this enough too. It's good at the early stage of development to start to initiate those API and drug product stability studies, so you get a feel, for how well your formulation is going to hold up, both through development, also ultimately through stability. And then when you get into the clinical side of things, your kind of focus needs to change a bit, where you start to need to think at, what is your sources of input materials going to be? Those input materials being, either your GMP lipids or your RNA itself. Starting to qualify those vendors and the potential of getting GMP grade material. And also, now that you've identified what those critical process parameters are, you want to look to further optimize them. And by optimizing, you want to ensure that your manufacturing process is going to be robust and repeatable, so that when you're doing at the clinical scale, you know what the quality of your drug product is going to be. And with those initial methods, analytical methods developed, you need to make sure those methods can be qualifiable for both. You have inputs into your system for the drug substance, and also methods for your final drug product. And then with that development, start to identify what those specific specifications are going to be. And then once you have a process that best mimics what your final manufacturing process to be, you're able to produce GLP tox batches. And finally, a lot of companies don't have their own manufacturing capabilities in-house, so necessary will be to transfer that process to a proper CMO, and then do the production as engineering and GMP batches in a clean room environment. And finally, have the QC system in place for the proper testing and release, and package and distribution, and start your official GMP stability program. So when we look at the total workflow here of what the manufacturing process is going to be, it's good to understand that, and then look at it in the flow chart format. And then you just have to start to identify what are all those input materials going to be when you're starting to look at how do you source them. So from a manufacturing standpoint, you have your nanoassembler, the particle production, and then dilution, and then also your tangential flow filtration steps. Adjust the concentration to where your final dosage is going to be, then sterile filtration, fill and finish. So if you look at different inputs into all these different unit operations, you have both your different buffers you're going to need and sources for that, and also your actual materials as well, whether it be the GMP microfluidic cartridges or your TFF cartridges, and sterile filtrations and products for refill and finish. But ultimately, with all of these kind of inputs into the system, as well as sourcing the materials, there's also a kind of a decision that goes into each one of these things. So you need to be able to justify the rationale behind each of these decisions, like why different buffers were used, why different lipid stocks were used, and that goes into your process development as well. So to have a good quality control system, as well as being able to identify the sources of different materials, you need to identify what the in-process testing is going to be and needs to be. So, obviously I mentioned this before, analytical is always going to be a big piece of product development. So I can't stress that enough, the importance of it. So, with your different in-process testing, like here's different examples here. And then it's important to have those different methods in place. And also it helps you show that you have good control of your manufacturing process. Different testing as far as measuring osmolality and pH of your buffers, measuring your RNA concentration, measuring your, you're typically using UPLC to measure the, verify the concentrations of your lipid stock, all the way down the line to your final, at the end of this will be your final methods for your drug product. And then along with your manufacturing process, there's going to be, as well as a total batch record to review the whole process. Each individual equipment will require its own SOPs. And now with the GMP NanoAssembler platform that we have, that comes with its own, full documentation package or SOPs around cleaning and operation, and also IQ, OQ. And then, with your TFF system, you'll need SOPs in place for that. And like I mentioned, all of that will be governed by a overall batch record. And a lot of times when you go to a CMO and do sterile filtration, they'll have their own batch record for the sterile filtration process. So we look at the GMP system itself. I mentioned some of these briefly, but as far as kind of enabling that regional and localized manufacturing, the system itself is comes as a standard system, but can also be configurable for fit for purpose for more specific applications. Gives you the capabilities. You have continuous flow up to in a 4-hour manufacturing window to get a 50-liter batch size. And it has a fully disposable fluid path, which takes away the need for generating cleaning validations. And it also can be integrated into your TFF system to make the overall process more continuous. And as I mentioned, it comes with a full documentation package to ensure to meet any necessary CGMP requirements, and also IQ, OQ documentations as well. So after the GMP system, you have your tangential flow filtration. So from a process development standpoint, there's obviously a lot of different parameters that can be incorporated with finalizing that, or I guess optimizing that TFF process. Here's a list of the different variables here. You can have different types of cartridges, different materials of cartridges, different molecular weight cutoffs, different surface areas. So really a lot of process development time can go into optimizing this process itself, which is why the Blaze instruments gives you the capability under R&D settings to optimize those parameters. And here's kind of a workflow of what with our own COVID development program, what that looks like, where you have your production on your GMP system, TFF concentration step, and then different buffer exchanges to both remove the ethanol and then, I said, finally put it in your final buffer for storage or cryo buffer. So one example here, so shear rate is one thing. I just want to give an example of something we looked at as a process parameter when developing our self-amplifying RNA-LNP vaccine. So shear rate is basically, ultimately it's directly correlated to the flow rate to your system. So a higher flow rate results in a higher shear rate. So just an example here is something we looked at. So we first looked at a 2.5 milligram scale. So production done on the Blaze, and then looked at different shear rates ranging from 4,000 to 8,000 inverse seconds. So what you see here ultimately is that, so at each step of the way from concentration to your two different diafiltration steps, here's your flux rates in parentheses. In kind of story here, without taking you all through this, is that by increasing the shear rate from 4,000 to 8,000 inverse seconds, you're able to reduce your total TFF processing time from 3 hours to under 2 hours while maintaining particle characteristics. And just as a note here, you typically use a TMP of 4 psi. And then, so this was at 2.5 milligram scale. And then to kind of, as you do scale up, you kind of see, so this is a 25 milligram scale on the actual GMP system. Looking at shear rates at 4,000 and 6,000, you start to see some even greater time savings and lower manufacturing time by increasing that shear rate. So this is an overall kind of workflow. These first two will be getting your materials ready, getting the system ready. And then finally here you see the runtime, 200 mils per minute. The runtime on the GMP system itself is extremely quick. So most of that time then is spent with your downstream processing and your TFF system. So what you see here ultimately is that by just slightly increasing the shear rate from 4,000 to 6,000 inverse seconds, you're able to reduce your processing time all the way from 7 hours to about 2.5 hours, which is a big benefit. So, and then looking at through development, looking at a few, in this case, 2 different LNP formulations, PDI LNP1 and LNP2. So this is an example where, having, as you develop your process, being able to sample along the way is critical too. So if there are any changes for your particle characteristics, you want to identify what step in the process those changes are happening. So here's, samples we're taking at different steps along the TFF process, after concentration, after diafiltration, and then after your final concentration, after filtration. So what you start to see here is with the 2 different formulations, LNP1, LNP2, you start to see a slight size increase with LNP1, enough, slight increase. So some calls for concern, but it doesn't tell you the full story. You see LNP2 here is consistent, and all of your percent encapsulation was consistent. But what you see here is when you then go and scale up this process from 2.5 milligrams to a 50 milligram scale, you see some of those slight differences get emphasized even more. So it's good to be able to go through the full process and really start to see where any of those process deviations could happen. So what you see here in this case is with LNP1, LNP2, you see the particle size of LNP1 starts to grow pretty substantially through your downstream purification process. However, you can also see here, because doing 2 formulations, you see LNP2 is able to maintain that those particle characteristics through that. So what the advantage of doing this and seeing this through development is, the LNP2 formulation was chosen for further scale up, and ultimately that formulation be used for the GLP tax and GMP manufacturing. And then with LNP2, you want to be able to demonstrate process robustness. So here you see a case here where that formulation was manufactured at clinical scale at 3 different sites. And what you see is you see the particle size and polydispersity is maintained throughout, and a slight variation when a different siRNA lot was used, but ultimately what this does show is process and formulation robustness. So finally, to summarize here, so if you go back and look at the whole process, so we have preclinical and clinical, different parts of the program, all available, that different services that P&I can offer as well. So you see with preclinical, that's where you're getting into your formulation process development. But then when you get into the clinical side of things, you're just starting to think of, how you're going to produce this at GMP or under GMP conditions. So we're starting to look at, sourcing of materials, where you're going to make these toxin GMP batches at. And then, we have developing the biomanufacturing capabilities to do that. From the analytical side of things, you're doing the initial development of those analytical methods. But on the clinical side of things, those all need to be regulated under a quality control system, and then developing a GMP testing and stability program. And also, something else we can also assist with is the RNA development and production as well. I'm going to stop sharing and open it up to the audience. So thank you, everybody.